As we are continuing with sessions in microbiology, please, if you haven't looked at a video on the culture medias, then on the morphologies of bacteria, please, you're requested to go back and look at them. So in this session, we are going to talk about a concept known as gram staining. This gram staining was developed in 1884 by a scientist known as Sir Hans Gram, and that's why we call it gram staining. So this one is being used to differentiate bacteria according to the Gram reaction or according to the staining reaction. And the staining reaction, we are going to look at the components of the Gram stain. We shall look at the principle, the procedure, and examples of Gram positive and Gram negative organisms. And if it is your first time on this channel, we request you to subscribe. That is the only pay you can give us, so that if whenever we release videos, you are able to be notified. And then please remember to like the video. So according to the components of gram stain, the gram stain contains what we call the primary stain. It has the primary stain. It has what we call a mordant. Then it has the decolorizer. Then a counter stain. So these are the four components of the gram stain. And this gram stain, the primary stain is known as crystal violet. The major one is crystal violet. But if you don't have crystal violet, you can use gentian violet, or methyl violet. So these are the three primary stains which you can use in a gram staining reaction, but the most preferred is the crystal violet. And these primary stains, if the organism is gram positive, it is going to retain the primary stain. So we are saying that gram positive organisms retain the primary stain, hence staining what we call purple color. That is what we can talk about the primary stain. Whereas the mordant, this mordant is used to fix. So the mordant is Rugose iodine, or you can call it Gram's iodine. And this iodine, the word mordant means when we apply the Rugose iodine, it is going to fix the primary stain in the peptidoglycan layer. For example, for the gram positive, this is for the cell wall of the gram positive, they have what we call a thick peptidoglycan layer. So if I apply the mordant known as Gram's iodine or Rugose iodine, it dehydrates the peptidoglycan layer, hence fixing the complex known as violet iodine complex. And when it is fixed on decolorization, it remains intact, hence the organism taking up the purple color. Then the decolorizer being used in the gram stain, we use acetone. Acetone is the most preferred component to be used as the decolorizer. So what does the decolorizer do? It is used that if the organism is gram positive, this decolorizer will not be able to remove the primary stain. Whereas if it is gram negative, the primary stain is removed. If it is gram negative bacteria, because of the presence of thin peptidoglycan layer and a layer of lipid which is dissolved by the alcohol. Hence, the gram-negative ends up taking the counter stain, which we call safranin. So after that is following after decolorization by acetone alcohol. Then the counter stain used in gram, the one of choice is safranin. Safranin can be used, but if you don't have safranin, you can use neutral red or dilute carbofucsin. Dilute carbofucsin, carbofucsin or fucsin. So these are the three counter stains which we can use. The primary stain, we have crystal violet, gentian violet, or methyl violet, which makes the gram-positive organisms look purple after resisting decolorization. Whereas the counter stains, we see safranin being the one of choice, but we can also use neutral red or dilute carbofucsin. So it means you get strong carbofucsin and you dilute it 1:20. It is always diluted 1:20. So that you can use it as also a counter stain. What is the importance of a counter stain? The counter stain, these ones, they enable or they impart color, which is they make the gram-negative to look pink or red. Why? Because gram-negative bacteria have a thin peptidoglycan layer. So when we apply a decolorizer, the violet iodine complex, the decolorizer dissolves the lipopolysaccharide layer on top, creating the pores in the peptidoglycan layer, and hence losing the purple color of violet. So when we apply a counter stain, these organisms take pink to red color. That is what we can talk about as a counter stain. So after looking at the components and their functions, now we want to look at the principle behind this. And the principle is already summarized here, whereby we are saying that gram-positive organisms have what we call a thick peptidoglycan layer. If you haven't watched our video on the cell wall of gram-positive versus gram-negative, please, you can visit it so that you get a bigger understanding of these words. So we have a peptidoglycan layer, and these gram-positive organisms, because they have a thick one, which is more polymerized. So this thick peptidoglycan layer, on adding what we call crystal violet, this crystal violet is able to enter inside the thick peptidoglycan layer. Then on addition of a mordant, that is the Rugose iodine, it is going to form a complex known as crystal violet iodine complex. And this crystal violet iodine complex is fixed within this thick peptidoglycan layer. And we said the use of the iodine as a mordant is to fix. It forms a complex which is fixed within the thick peptidoglycan layer. So on addition of the decolorizer, that is acetone, the crystal violet iodine complex, what is the acetone going to do? It is going to dehydrate. It is going to dehydrate the thick peptidoglycan layer, causing the crystal violet iodine complex to be fixed within the thick peptidoglycan layer. Hence, the organisms are going to retain what we call the purple color. The purple color is going to be retained, which is different from gram-negative. For the gram-negative organisms, for them, they have what we call a very thin peptidoglycan layer. As we are seeing it here, because this is for gram positive, this is for gram negative. So for gram negative, they have a very thin, a single layered peptidoglycan with polymers and liposaccharide. So when we apply a mordant, that is iodine, it is going to form crystal violet iodine complex. But this complex is not retained on addition of a decolorizer. So when we add acetone, this acetone is first of all going to dissolve the lipid layer. It dissolves the lipid layer, or what we are calling the lipopolysaccharide layer, on top of the peptidoglycan. After dissolving it away, it is going to create polynes, or what we are calling pores, hence allowing removal of the counter stain. So the counter stain is removed. So on decolorization for 5 to 10 seconds, the purple color, which we are calling the primary stain, is removed. So the organisms do not remain purple. So on application of the counter stain such as safranin, so when we add safranin or neutral red or dilute carbofuchsin, these gram-negative organisms are going to take up pink or red color as seen in microscope. And these ones we shall say these are gram-negative. Whereas those ones that take up purple color, they are known as to be gram-positive. And the reason is on the size of the cell wall, the peptidoglycan layer. In the gram-positive being thick, whereas in the gram-negative having the lipid layer with the pores at the same time with a thin peptidoglycan layer, which does not allow the violet iodine complex to remain on the organisms. Hence, they are lost on decolorization and ends up taking the counter stain. So after looking at the principle, now let us look at the procedure. Hope you guys you're following very well. And with the procedure is that we have our requirements whereby we need the primary stain, that is we are going to use the crystal violet as the counter stain. We have the crystal violet, and it is always blue in color or purple. Then we have what we call the Rugose iodine. Iodine, I'm going to denote it in black color. This is grams or Rugose iodine. Then we have the decolorizer. Since it is always colorless, I'm going to leave it like this. Then we shall have a counter stain known as saffron, which we are going to use. So this counter stain for it is red in color. So these are the requirements we need to have under gram staining. We need a staining rack. We need the heat to fix, or what we call a source of heat. So what we do, if we have already made film, the first thing we do is to heat fix. You heat fix the smear for like 1 or 30 seconds or 1 minute. So we do heat fixing, that is you assemble requirements, then you heat fix the smear. After fixing the smear, you come and you stain it with, you cover it. We use the word cover. You don't flood the smear, but you cover the smear with crystal violet. So what we are going to do, we're going to come and cover this smear with the crystal violet stain. And after covering it with the crystal violet, we leave it to stain for 1 minute. After 1 minute, we wash off the crystal violet and we apply what we call a mordant. So if we had our smear here, so we come and apply a mordant, what we call Rugose iodine. We apply it, Rugose iodine is in black. Remember the smear at first is already bluish, sort of bluish. But after washing, we come and apply the rugose iodine, and we leave it on the smear also for 1 minute. 1 minute. After 1 minute, we are going to still wash with running tap water. We can use either distilled water or tap water. After washing off excess of the stain, we go and decolorize. And on adding a decolorizer, that is acetone. The decolorizer we are using is acetone. So if we had our smear with rugose iodine with a mixture of some of the remnants of the crystal violet, we are going to see it being decolorized. So at the end of adding acetone, and we stain it for only 10 seconds, only 10 seconds. So when we apply the decolorizer, at the end of decolorizing, the smear is going to lose, or it is going to remove all the bluish color. If it is gram negative, if it is gram positive, it is going to retain some little of the color. So that is decolorizing. So it retains small color of the crystal violet. That is for 5 to 10 seconds. After 10 seconds, we wash. Then after washing, we apply a counter stain. That is safranin or neutral red or dilute carbofuchsin. Then after adding this, if we had a smear, after adding this safranin, it is going to appear reddish. So for the gram negative, if they had lost this crystal violet, they are going to take up the counter stain, and even it is on timed for 1 minute. Then after this, we wash, and then we end by air drying. You air dry the smear, and it is ready to examine. So when you examine microscopically the smear after air drying, we examine using times 100 objective lens. And when we examine using times 100 objective lens, we are going to look to find that for gram-positive organisms, if this is the field, for the gram-positive, they are going to appear as purple. If they were cocci, they are going to exist as purple cocci. And if it is gram-negative, if this is the field and we are examining under times 100, we are going to find that they are red, they are reddish in color. Either they can be bacilli, which are red, or cocci, which are taking up pink or red color. So this is how they are able to appear under microscope. This one is a gram positive, these are gram negative. Okay, so what are the examples of gram-positive organisms? Examples include, under the cocci, we have examples like Staphylococcus, we have Streptococcus species, we have the Enterococcus species. These are the examples of the cocci which are gram-positive. Staphylococcus being in chain, in cluster, Streptococcus being in chain. Then we also have the bacilli, radio. We have the bacilli, and the bacilli examples we have include the Corynebacteria species. We have the lactobacilli. We have the Mycobacterium tuberculosis, which causes TB. We have Another example as Clostridium species. Clostridium species. These are examples of bacilli which are gram-positive. Then the cocci which are gram-positive, we have Staphylococcus, Streptococcus, and Enterococcus. Then examples of gram-negative. Here under gram-negative, we have under the cocci, those are ones which are oval in shape. We have Neisseria species. Neisseria species such as Neisseria gonorrhoeae, which is a gram-negative diplococci. Then we have different bacilli, or what you can call loads, such as the Enterobacteriaceae. And there are many examples of Enterobacteriaceae, whereby under Enterobacteriaceae we have organisms like Salmonella, like Shigella, like Proteus, Pseudomonas. All these are gram-negative loads. Loads meaning they are like a load. Then these ones, cocci, they are always oval in shape. And they stain what we call a red color. Not forgetting organisms also here like Brucella. Brucella is also gram-negative. Organisms like Vibrio cholerae are also gram-negative. On gram-positive, we can even add an organism known as Nocardia. Is also gram-positive. So all these are gram-negative, whereas these ones are gram-positive. Then we have also those ones which are gram-variable, like actinomycetes. So organisms like actinomycetes, for them they are gram-variable. These ones are gram-variable. They can appear as an intermediate of both gram-positive and gram-negative. So this is what we wanted to discuss with you in this session, where we have looked at the components of the gram stain, that is the primary stain, the mordant, the decolorizer, and the counter stain. We have looked at the principle whereby it is due to the size of the cell wall, whereby the gram-negative positive have the thick, whereas gram-negative have the thin peptidoglycan layer. We have looked at the principle, the procedure whereby we can use a mnemonic known as come in and stain. You can use the mnemonic come in and stain, whereby come is crystal violet, in is iodine, acetone, and safranin. Then we have looked at different examples. Thank you so much for learning with us. If you have enjoyed the video, please remember to like the video. Share to your friends so that they can continue to learn with us. And if you haven't subscribed, please subscribe. May God bless you.